Hello everyone, my name is Rahul. In the previous video, we had discussed about Bt cotton. In this video, we are going to discuss about nematode resistant plants. But before we move on to nematode resistant plants, let us first understand what is RNA interference. RNA interference, it is a novel method which involves silencing a specific mRNA, silencing a specific mRNA such that this mRNA cannot be translated, and therefore no protein will be produced. So we know the central dogma of life, that DNA is transcribed to form mRNA and mRNA is translated to form protein. Now RNA interference means that we have to disrupt this translation process such that this protein will not be formed. Now RNA interference uses a double-stranded RNA or a dsRNA for gene silencing. Gene silencing means that this gene will be transcribed to form an mRNA, but it will not be translated to form a protein. Now, how is this achieved? It is a very complex process. So we will see a very simplified version of this complex process, because this you can read in higher classes also, but for now this simplified version will suffice. So what we are basically doing is we take a double-stranded RNA, by the help of a protein known as dicer. A protein known as dicer can be converted into three forms. First is known as siRNA, the other is known as shRNA, and the third one is known as miRNA. siRNA stands for small interference RNA, sh stands for small hairpin RNA, and mi stands for microRNA. These siRNA, shRNA, miRNA, along with certain proteins and enzymes which are known as splicer and argonaute, they are involved in gene silencing by formation of RNA interference silencing complex known as RISC, RNA interference silencing complex. This RNA interference silencing complex, it binds to the target mRNA, and it leads to fragmentation of the target mRNA, and thus no protein is produced. For example, if this is a cell, in this cell, this is my double-stranded DNA. It will be transcribed to form a single-stranded mRNA. Now what I do is I introduce a double-stranded RNA. Now this double-stranded RNA, by the help of these above mentioned enzymes, dicer, argonaute, and dicer, leads to the formation of RISC, RNA interference silencing complex. So what this RNA interference silencing complex will do is it will convert this double-stranded RNA into small fragments. Now, these small fragments of double-stranded RNA, they will be compatible, they will be complementary to this target mRNA. So these small fragments will bind to this target mRNA like this and will lead to fragmentation of this target mRNA, and this is how this particular gene will be silenced. So this method involves silencing of a specific mRNA due to a complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA, which is called as silencing. Now, the source of this complementary RNA, the complementary double-stranded RNA, it could be from an infection by plasmids or viruses which contain these RNA genomes, or it could be through transposons. So, source of this double-stranded RNA, it could be viruses with RNA genome which are able to produce these double-stranded RNA, or it could be transposons. Transposons are also known as mobile elements, are also known as jumping genes. Now we know that in a chromosome, the location of each gene is fixed. The location of each gene is fixed. So this is a gene, and its location will be fixed. Now transposons, they are special type of mobile elements that can change their position in the chromosome. So these transposons or mobile elements, they can be used to deliver this double-stranded RNA to the required cell, and this will act as a source of this double-stranded RNA by this machinery of the dicer, splicer, argonaut, etc. It will be converted into RNA interference silencing complex and will ultimately lead to fragmentation of target mRNA, and thus no protein will be produced. Now, this RNA silencing, this RNA interference, it takes place, RNA interference takes place in all eukaryotes, as a method of defense. It takes place in all eukaryotes as a method of defense. So what we do is we only need to transfer this double-stranded RNA, and the rest, that is the conversion to the RNA interference silencing complex, will be done inside the cell because it is common to all eukaryotes. All eukaryotes already have these dicer, slicer, and other enzymes which are required for this RNA interference. Now, this is the tobacco plant which has been affected by a nematode. These nodules, they show infection by a nematode, and this shows infection in a nematode resistant plant which has been developed by the technique of RNA interference. So there is a nematode, which is known as Meloidigyne incognitia. Which infects roots of tobacco plant, and this causes great reduction in yield and increased losses. So a novel strategy was adopted to prevent this infestation, which was based on RNA interference using agrobacterium vectors. We introduced nematode specific genes into the host plant. The introduction of DNA or the genes was such that the introduction of DNA or specific genes was such that it produced both sense and antisense, RNA in host cells. Now normally what happens is, suppose this is a DNA molecule, a double-stranded DNA molecule. When it is transcribed, only a single-stranded mRNA is produced. Now we introduced the vectors, agrobacterium vectors, with the genes which were specific to nematodes, such that a double-stranded RNA was produced. This double-stranded RNA, one strand is known as the sense strand and the other is known as the anti-sense strand. Since they are both complementary to each other and they form bonds, this mRNA is non-functional. That is, it will not be transcribed, it will not be translated to produce proteins. Now this sense and antisense RNA, they form a double-stranded, they form a double-stranded RNA. As soon as a double-stranded RNA is formed, it initiates RNA interference. And how it takes place, we have already seen it. It initiates RNA interference and thus it silences the specific mRNA of nematode against which we have targeted this DNA or the double-stranded DNA or the double-stranded RNA. So the specific gene of the nematode will be silenced and therefore nematode will not be able to survive. So that is how this transgenic plant will protect itself against this parasite. So this is how RNA interference takes place. With this, we finish off with this video on nematode resistant plants. See you in next video.